All right, so today we're going to we're going to start by looking at d orbitals, okay, in transition elements and the shapes of different d orbitals, and then we'll go on to explain why transition metal complexes are colored, okay, or why they form colored solutions or colored compounds, transition elements. So we're going to start with d orbitals, okay. Now there are a total of 5 d orbitals, okay. We're going to be looking at the 3d orbitals. And you can view the d orbitals as sort of two different sets. Okay, you have orbitals between the axes, okay, orbitals between axes, okay, and you have orbitals along axes, okay, and I'm just going to show you how we can draw both, okay, orbitals along the axes. Now we have three different types of orbitals between the axes, okay, we have three different types of orbitals between the axes, okay, so I'm going to draw the x, y, and z axes, right, so let's say this is my y-axis, this is my x-axis, and then this is my z-axis, okay. Now it doesn't matter which axis you show where, okay, it doesn't matter if you show the z vertically, for example, or you know the y-axis vertically. So the first type of orbital that we have, is called a dxy orbital, okay. The dxy orbital is found between the x and y axes, okay, it's found between the x and y axes, and it is a double lobed orbital, okay. So it's sort of like a double p orbital, okay, like this. The next d orbital that we have is the dxz orbital, okay, dxz. Now the dxz is between the x and z axes, okay. Now again, as I said, it doesn't matter how you label the axes, okay, as long as you show it between the relevant axes. So if you have to draw the dxz, just label the vertical axis, for example, as the z axis, and label the horizontal axis as the x-axis, okay. That way you can always label the axis that you don't want to show the orbital on as the axis that is going from back to front, okay, or front to back, right. So that would be your z-axis, and that would be your y-axis in this case, right. So the dxz orbital will be drawn between the x and z axes, okay. Okay, it will be drawn between the x and z axes. The third type of orbital that we have between the axes is called the dyz orbital, okay. Now for the dyz orbital, you can simply show, let's say, the vertical axis as the z-axis and the horizontal axis, let's say, as the y-axis. Then we're going to show this axis as the x-axis, okay. And the dyz orbital will again be a double-lobed orbital between the y and z axes, okay. It will be between the y and z axes, right. So you have this, and then you have this. So if you're asked to draw the axes, okay, it doesn't matter which you label as the x, y, or z axis. So it's just easier to simply label the axes that you want to draw the orbital for, right, on the plane of the paper, right. And the third axis can be the one shown going front to back. Okay, but if they have already drawn the axes for you and labeled them, then make sure that you draw the orbital such that you've shown them between those two axes, okay. The dxy is between the x and y axes, the dxz is between the x and z axes, and dyz is between the y and z axes. Now we're going to look at the orbitals along the axes, okay. So there are two types of orbitals that we have along the axes, right. The two types of orbitals that we have along the axes are called the dx squared minus y squared and the dz squared orbitals, okay. Dx squared minus y squared and dz squared. Now the dx squared minus y squared is along the x and y axes, okay. It is along the x and y axes. So along the y axis and along the x axis, okay. Along the x axis and y axis is the dx squared minus y squared orbital, okay. And then the dz squared is along the z axis, okay. The dz squared is along the z axis, right. So you have this, and then you have sort of this ring, okay, this ring or donut around that around that double lobed shape. So we know that the D subshell, right? If you look at the 3D subshell, for example, or any D subshell, we have how many different orbitals? We have 5D. We have 5 different D orbitals in the D subshell, right? 3 of those orbitals are between the axes, and then 2 of those orbitals are along the axes. Now, all 5 of these D orbitals, okay, all 5 of these D orbitals belong to the D subshell, right? And all of them are at the same energy, right? We've learned that in electronic configuration, right? So if I have, let's say I have a copper 2 plus ion, right? In my copper 2 plus, my configuration for copper is argon 3d10 4s1, right? Which means that my configuration for copper 2 plus is argon and 3d9, right? Now, so this is my electronic configuration for the copper 2 plus ion, right? And what I know is that if I look at the d orbitals, okay, if I look at the d orbitals, right, here I have energy on the y-axis, okay, and all 5d orbitals are at the same energy, right, within the same subshell, right, within the d subshell. These are called degenerate d orbitals, okay. These are called degenerate d orbitals. Okay. The word degenerate over here means orbitals at the same energy level. Okay. The word degenerate means, okay, so when we talk about degenerate orbitals, okay, they are orbitals at the same energy. Okay. They are orbitals at the same energy. So if we had something like an isolated copper atom, we know that all 5d orbitals in the d subshell are at the same energy. So those d orbitals are called degenerate d orbitals. Okay, all 5d orbitals are at the same energy. Now let's say that I take copper 2 sulfate. Okay, I take copper 2 sulfate and I dissolve it in water to make an aqueous solution of copper 2 sulfate. Okay, and we know that in this aqueous solution we have the following complex ion, right? That is Cu H2O 6, right, 2 plus. Okay, this complex is called the copper 2 hexa aqua complex. Okay, it is called the copper 2 hexa aqua complex. Okay, and the question is, why is this complex colored? Okay, or why is this solution colored? Copper 2 hexa aqua. Okay, the 2 refers to the oxidation state of copper. Hexa means 6, and aqua is a reference to water. Okay, so it's copper 2 hexa aqua, right? And we know that this is a pale blue solution, right? It is a pale blue solution. The question is, why is it a pale blue solution? Now over here, if we just had an isolated copper 2 plus ion, right? In the D subshell we have 9 electrons, right? In the D subshell we just have 9 electrons, right? So the copper 2 plus ion has 9 electrons in the 3D subshell, right? And all of those orbitals are at the same energy. They're called degenerate d orbitals. Okay. Now what happens is that when the ligand, in this case water, right, it's a monodentate ligand, right, when these ligands start bonding to the copper 2 plus ions, right, they form dative bonds. They donate a lone pair of electrons to form dative covalent bonds to the central metal ion in this case. What happens is that these orbitals experience repulsion from the incoming lone pairs of the ligands. Okay, these orbitals, okay, or these electrons experience repulsion from the incoming electrons, right? Those lone pairs that you get from the ligands. So what we can say is that the lone pairs, okay, the lone pairs of electrons, right? Lone pairs of electrons from ligands, okay, repel the 3D electrons. Okay, repel the 3D electrons. And in an octahedral complex, we know that the ligands bond along the axes. Right? So because the ligands bond along the axes, what that means is that What that means is that the orbitals along the axes are repelled more than the orbitals between the axes. So the two orbitals along the axes are repelled to a higher energy relative to the three orbitals that we have between the axes. So again, in an octahedral complex, okay, in an octahedral complex, the ligands are forming bonds along the x, y, and z axes, which means that the orbitals along the axes, which were your dx squared minus y squared and dz squared, right? Dx squared minus y squared and dz squared, right? These two orbitals are repelled more than the orbitals between the axes. So the dxy, the dxz, and the dyz remain at a relatively lower energy. So over here, right, I can say that these are my 3D orbitals. These are my degenerate 3D orbitals, okay, in a copper 2 plus ion, right? Whereas in my octahedral complex, right, my d orbitals, right, in the copper 2 ion now look like this. They are split into two different sets with different energies. And now my 9 electrons aren't all at the same energy, right? I have some at a lower energy and some at a higher energy. So sorry, this should be the YZ, right? Not XZ. This is YZ. Right? So we have DXZ, DXY, DXZ, and then DYZ, right? Those are my three orbitals. Those are my three different d orbitals between the axes, right? So again, those are at a relatively lower energy, and the orbitals along the axes are repelled to a higher energy because the ligands bond along the axis in an octahedral complex. Now these d orbitals are what we call non-degenerate d orbitals, right? They are called non-degenerate d orbitals, right? Degenerate d orbitals are d orbitals at the same energy. Non-degenerate d orbitals would be orbitals at now within the same d subshell, right? Three of the orbitals are at a lower energy and then two of them are at a higher energy, right? So they're non-degenerate. Not all five are not at the same energy. So now let's try and answer the question as to why transition element complexes are colored. So for example, why does this copper 2 hexaaqua complex, right? Why does it form a pale blue solution? So now let's try and understand why these complex ions form colored solutions, okay? Or why solutions of these complexes are colored, okay? Now, for example, right? Let's say that we have a solution, okay? So that we have a solution of copper 2 sulfate aqueous, right? Right, and in this solution, we know that copper ions exist as copper 2 hexa aqua. Right? Again, in this aqueous solution, they do not exist freely as copper 2 plus ions. Okay, these copper 2 plus ions end up forming this octahedral complex, okay, in aqueous solution. And we know that this octahedral complex, instead of the orbitals being at the same energy level, those are degenerate d orbitals. The d orbitals are at different energies. So the energy, you can say that the d orbitals are split into two levels of different energies, right? The orbitals along the axis at a higher energy, the orbitals between the axis at a lower energy. Okay, so again, these are called non-degenerate d orbitals within the same subshell, but not at the same energy. Now, the reason why this solution, okay, the reason why the solution is colored is because let's say that white light is passing through the solution, right? So this solution is sitting outside, or it's sitting in a lab, for example, right? Natural light is white light, okay, and white light, you can think of white light as the visible spectrum, right, which is VIBGYOR, right? Violet, indigo, blue, green, yellow, orange, red, right? So these are the colors. These are the colors that we have in white light, right? The visible spectrum. White light can be thought of as a mixture of all of these colors. Now, as white light passes through the solution, what happens is that again, white light is a mixture of all of these colors. Okay, white light is a mixture of all of these colors, and a specific colors are absorbed by the complex ion. Okay, a specific frequency of light. Okay, a specific frequency of light is absorbed by the is absorbed by the solution. Now, how does that work? Okay, so over here, let's say that the difference in energy between the lower energy d orbitals and the higher energy d orbitals is delta E. Okay, so delta E is the difference in energy, okay, between the two sets of d orbitals. Okay, and we know that energy is directly proportional to frequency. Okay, so in this particular case, this difference in energy, okay, will correspond to a particular frequency of visible light. Okay, this difference in energy will correspond to a particular frequency of visible light. Okay, now again, this difference in energy is corresponds to a very specific frequency of visible light. Okay, and that frequency of visible light, when it's absorbed by the solution, okay, it results in, okay, it's absorbed, resulting in the promotion, okay, resulting in the promotion or excitation of an electron from a lower energy d orbital to a higher energy d orbital. So what happens is, again, you're passing white light through the solution, right? White light is a mixture of all of these different colors, right? Each, obviously, every color in the visible spectrum corresponds to a particular frequency of or wavelength, right? Frequency or wavelength. In this case, we're looking at frequency. So a specific frequency, in other words, a specific color is, so a specific frequency of visible light, or in other words, a specific color is absorbed, okay, resulting in the excitation or promotion of an electron from a lower energy d orbital to a higher energy d orbital. Okay, and what we see is what is observed is the complementary color. Okay, what we observed, what we observe here is called the complementary color. Okay, so in this case, the complementary color that we're observing is pale blue. Okay, in this case, the complementary color that we're observing is pale blue, right? That's the copper 2. That's the copper 2 sulfate solutions color, right? So that is your pale blue solution in this particular example. We have a solution, right? What's happening is that solution is sitting in natural light. Natural light is white light. It's a mixture of all the different colors. It's a mixture of all the different colors. A specific color is absorbed. Essentially, you can think of a specific color essentially stays back in the solution, and the remaining colors are what you observe. So if you have, you know, so for example, whatever color is absorbed, you can think of it as staying in the solution, and the mixture of the remaining colors is observed, and that complementary color is, in this particular case, is pale blue. Okay, so it's like white light minus the color that was absorbed. Okay, it's white light. It's a mixture of all the remaining colors. That's going to be the complementary color. So for example, if we take the example of plants, right, or leaves are colored green, right? Plants are green. Now, what happens in plants is that, for those of you that take biology, you would know that plants have photosystems, okay? But plants basically absorb a specific frequency of visible light, okay? Specifically, they absorb blue light and red light. They absorb blue light and red light, and what you end up observing is the color green, okay? So in the case of plants, blue light and red light essentially are absorbed by the plant, whereas the color that you observe is the complementary color, that is green. Okay, in this case, a specific frequency was absorbed, and you observed the complementary color, which was pale blue. So what we can say over here is the following. Okay, we can say, if the question is asked, why are transition element complexes colored? Okay, so why are complexes colored, or why do they often form colored solutions? Okay, we can say that when a complex ion forms, okay, the d orbitals, okay, the d orbitals are split. The d orbitals are split into two different sets. Okay, into different sets. The orbitals are split into different sets with different energies. Okay, different energies. The difference in energy, okay, the difference in energy delta E. Okay, you can use the abbreviation delta E. Okay, the difference in energy between the two sets of d orbitals corresponds to a certain frequency of visible light that is absorbed resulting in okay, resulting in promotion or excitation of an electron from lower energy d orbital, okay, lower energy d orbital to higher energy d orbital, lower energy d orbital to higher energy d orbital. Okay, and because a specific frequency of visible light is absorbed, right, the remaining frequencies are the color that we observe. Okay, it's the mixture of the remaining frequencies is the observed color. Okay, so we can say that the complementary color is observed. Okay, the complementary color is observed. The complementary color is observed. Right, so again the d orbitals are split into different sets, okay, with different energy. So we have to reference to the splitting of d orbitals. Okay, the d orbitals are split into different sets with different energies. Okay, then we know that the difference in energy corresponds to a certain frequency of visible light. Okay, we have to specify that certain frequency of visible light, okay, that is absorbed, resulting in the excitation of an electron, okay, from lower energy to higher energy d orbital, and then the complementary color is observed. Okay, sometimes for octahedral complexes you will see the abbreviation O subscript D used, okay, in the marking scheme. All right, octahedral complexes are sometimes donated by this abbreviation for their geometry. All right, O subscript lowercase d. Right. Now then the question arises, why do different complexes have different colors? Okay, the first thing to understand is that it is not the copper 2 ion that is responsible for this color. Okay, copper 2 plus ions in aqueous solution can have different colors. Okay, we've learned that copper 2 is a pale blue solution, but that is because we formed the copper 2 hexa aqua. Okay, in other aqueous solutions copper 2 might have a different color. So for example, if I take this solution, and I add concentrated hydrochloric acid to it, okay, this copper hexa aqua, will convert into what we call copper tetrachloride. Okay, this copper hexa aqua will convert into copper tetrachloride. The copper 2 hexa aqua, when it forms copper 2 tetrachloride, okay, copper 2 tetrachloride is a yellow solution. Okay. The CuCl4 2 minus, okay, is a yellow solution. Again, it's the same copper 2 plus ion. It's the same metal ion here, right? But it's a different complex ion. So it's not the metal ion that determines the color, it is the complex ion. Okay, different complexes have different colors. So the question arises, why do different complexes, okay, why do different complexes, okay, have different colors? Why do different complexes have different colors? Now the reason different complexes have different colors is because, okay, the difference in energy delta E, okay, between the non-degenerate, d orbitals can be different. Okay, between the non-degenerate d orbitals is different in different complexes, right? In different complexes, the difference in energy between those two different sets of d orbitals is different, right? Which means that we can say that if the difference in energy between the d orbitals is different, right, we can say therefore, right, different frequencies of different frequency of visible light will be absorbed. Different frequencies of visible light will be absorbed, and therefore different complementary colors will be observed. Okay, hence different complementary colors will be observed. The complementary colors are observed. Now in tetrahedral complexes, right, we know that copper 2 tetrachloride is a tetrahedral complex ion. Okay, in tetrahedral complexes, the splitting pattern is the opposite of what you had with octahedral. In octahedral, the orbitals along the axes were repelled to a higher energy, okay, and the orbitals between were at a lower energy. In the case of tetrahedral, it's the opposite. Okay, the orbitals between the axes are repelled to a relatively higher energy, whereas the orbitals along the axis are at a lower energy. Okay, so what you get here is at a lower energy you have dx squared minus y squared and you have dz squared, right? These two d orbitals, and at a higher energy you have the dxy orbital, the dxz, and the dyz. Okay, so that is the splitting pattern in the tetrahedral geometry is different. Okay, so here you have the splitting pattern for copper 2 plus in tetrahedral geometry. Okay, sometimes this will be abbreviated as the following. Okay, it will be capital T subscript D. So again, in the isolated ion, the orbitals are degenerate, whereas in the complex ion, the orbitals are non-degenerate, and the from complex to complex, the difference in energy can vary. Okay, from complex to complex, the difference in energy can vary, and if the difference in energy varies, that means the frequency of light, visible light absorbed, varies, and therefore the complementary colors that you observe also differ. Okay, the splitting pattern. Okay, the splitting pattern is it depends on the geometry. Okay, so the geometry determines the splitting pattern. Okay, whereas the color is dependent on the difference in energy. Okay, now what geometry a complex has is determined by the ligands it's bonded to. Okay, and the splitting pattern also is affected by the difference in energy is also affected by the ligands. Okay, but they're two different things. You can have two things with similar geometry that have very different colors, right? You can have two things with different geometries that have very similar colors as well. Okay, the color is only dependent on what the difference in energy is, whereas the splitting pattern is determined by the geometry of the complex form. Again, what we should note is that when the d orbitals, the degenerate d orbitals, are split into non-degenerate orbitals, okay, the non-degenerate orbitals, both sets are higher in energy than the original, degenerate d orbitals, okay, because of repulsion due to presence of ligands. Is it that some are repelled more than others? Okay, so again, these five will be lower in energy. In the degenerate state, or when they were at the same energy, they were lower in energy, and when they formed complex ions, all five d orbitals were repelled to a higher energy, whereas but some were repelled more than others. Okay, the same in octahedral. Okay, in octahedral complexes, right, the ones along the axis are repelled more than the ones between the axes. Okay, but they're both sets are higher than in energy than the original degenerate d orbitals. Okay, whereas for tetrahedral, it'll be the ones between the axes that are repelled more than the ones along the axes. Okay, so again, both sets will be higher than the original set of d orbitals, but some will be repelled more than others. The last thing I want to talk about is the color wheel. Okay, you can essentially use this color wheel to determine the color of a complex ion. So here I have the color wheel. Okay, now for the color wheel we have the following colors. Okay, we have purple. Okay, then we have blue. Okay, then we have light blue. Okay, light blue or cyan. Okay, cyan or light blue. Okay, then we have green. Okay, we have green. Then we have yellow. Okay, and then we have red. Okay, so this is the color wheel that we have. Okay, and we can use the color wheel to determine the color of a complex ion. So for example, we know that the copper 2 hexa aqua ion, right, has sort of a pale blue color. Okay, now the absorption, okay, this particular complex absorbs in the yellow, okay, absorbs in the yellow and red region, right? It absorbs in the yellow and red region. So if you look at the color wheel, right, if it absorbs in the yellow and red region, the complementary color is the one directly opposite that. If it absorbs in yellow and red, we can see that directly opposite yellow and red is blue and light blue, and that's the color that we observe, right? So the complementary color, right, which is the observed color in this case is what? Pale blue, right? The complementary color or the observed color in this case is pale blue. Okay, so if we are given an absorption spectrum, if we are told which colors are being absorbed, okay, or which frequencies of visible light are being absorbed, we can determine the complementary color. Okay, it will be directly opposite the frequencies that are being absorbed in this color wheel. Another example that we have is Cr H2O6, okay, 3 plus. This is the chromium 3 hexa aqua complex, okay, and this absorbs, okay, it absorbs in the blue and red region, okay, it absorbs in the blue and red region. Okay, absorbs in the blue and red region. So if it's absorbing in the red and blue region, right, so they're sort of in this region, red and blue, the color directly opposite that is green. So the complementary color, that is the observed color in this case, okay, the complementary color, right, that is the color we're going to observe, that's the color of the solution, okay, will be green. Okay, complementary color here will be green. So the complementary color here is green, and we know that chromium 3 plus in aqueous solution, right, has a green solution, right? So this is how we can determine the color of a solution based on the absorption data given to us. Okay, if they tell us what regions the absorption is taking place in, we can identify the color of the solution by looking at the complementary color that is directly opposite the absorbed colors. In the case of copper 2 plus ions, they absorb in this sort of orange region between red and yellow. So the observed color is somewhere between light blue and blue. Okay, it's somewhere between light blue and blue. Okay, similarly, chromium 3 plus aqueous solution, absorption is in the red and blue region. So the observed color is directly opposite that. That's the color of the solution. That's the complementary color. That is going to be green.